Welcome back to the deep dive. Today we are wading into some pretty murky waters, literally.
We are. We're leaving the clean, predictable environment of the hospital and heading out to the coast.
Exactly. To the estuaries, the brackish waters where microbiology really meets marine biology. We are unpacking Chapter 26 of Murray's Medical Microbiology, 9th edition.
And it's a chapter that I think really makes you look at bacteria differently. We tend to think of pathogens as these, dedicated invaders that just want to harm us.
Right. Their sole purpose is to make us sick.
But the organisms we're talking about today, Vibrio and its relatives, they're environmental survivors first and foremost. They don't need us. We're just an unfortunate and often deadly accident in their life cycle.
That is a humbling way to put it. And to make sure we've got all our bases covered, we'll also be pulling in some key identification details from Jowitz just to lock down the diagnostics.
A good call.
So the topic is Vibrio and related bacteria. I've been thinking of them as the curved rods of the coast.
That's a pretty good name for them. Accurate and maybe a little ominous.
Well, it fits. We have a huge amount to get through. We're going to touch on the history-shaping power of Vibrio cholerae, the Blue Death. We'll look at those nasty infections you can get from bad sushi with Vibrio parahaemolyticus.
And we have to confront one of the most aggressive pathogens out there, Vibrio vulnificus.
Oh, absolutely. Plus, we can't forget their freshwater cousin, Aeromonas, the great mimic.
Definitely not.
Before we get completely lost in the taxonomy, though, I want to frame this for everyone listening. If you need one single mental model for this whole deep dive, it's this triad. Water is the vector, toxin is the weapon, and dehydration is the killer.
That's perfect. That triad really nails the clinical picture, especially for cholera. It's such a unique disease because it's a disease of pure physiology.
What do you mean by that?
The bacteria isn't destroying your gut lining like, say, Shigella. It's not invading your bloodstream. It just sits there and flicks a tiny chemical switch that turns your body's own cells against you. It turns you into a faucet.
A faucet. I was thinking more like a fire hose.
Okay, fair point. Fire hose. And the real tragedy of it is that the pathology is completely reversible. If you understand the electrolyte balance and can replace what's being lost, you can save the patient.
But if you don't?
They can die in a matter of hours. It's genuinely one of the fastest killers in medical history.
All right, let's get into the nitty-gritty then. Section 1. Physiology and structure. We need to know who these characters are. Murray starts by calling them comma-shaped rods.
Right. And this is our first big taxonomic point. Historically, Vibrio and Aeromonas were sort of lumped together because under a microscope, they look pretty similar. Both are gram-negative rods. Both can cause GI issues.
But genetically, they're different.
Exactly. Murray is clear on this. Vibrio is in the Vibrionaceae family, and Aeromonas now has its own family, Aromonidaceae. They're neighbors, not siblings. But for us, clinically, we group them together because the diagnostic workup starts the same way.
Okay, so let's play that out. You have a patient with acute water diarrhea. You get a stool sample, do a gram stain, and you see gram-negative rods. Your mind immediately goes to E. coli salmonella. How do you instantly know you might be dealing with something different?
This is your first major exam anchor. The fork in the road. It's the oxidase test.
The oxidase test.
All of the Enterobacteriaceae, that's your E. coli, Klebsiella, Salmonella, Shigella, all of them, are oxidase negative.
Okay, the whole family. Oxidase negative.
But Vibrio and Aeromonas are oxidase positive.
Let's just pause on that. What are we actually testing for? I think a lot of us just memorize positive or negative without really knowing what the test is doing.
That's a great point. The oxidase test is looking for a specific enzyme, cytochrome C-oxidase. It's a component of the electron transport chain that some bacteria use for respiration.
So it's about how they breathe, essentially.
In a way, yes. The test region is a chemical that will donate an electron to that enzyme. If the enzyme is present, it grabs the electron, and the reagent changes color, usually to a deep purple or blue.
So purple is positive. Purple means it could be Vibrio or Aeromonas. No color change means you're probably looking at something like E. coli.
Precisely. In a test question, if you see oxidase positive next to a diarrheal pathogen, you can immediately strike through that whole list of Enterobacteriaceae. It narrows your differential diagnosis in a heartbeat.
Okay, so they're gram-negative, oxidase-positive rods, but you said they're not just straight rods.
No, they're curved. Comma-shaped is the classic description. And they're facultatively anaerobic, so they're flexible, they can use oxygen, or they can ferment. But the other huge feature is their motility.
They move fast, I remember that.
Incredibly fast. They have polar flagella. That means they have a flagellum, like a tail, at one or both ends of the cell.
As opposed to being covered in them.
Right, which would be paratrichous flagella, like you see on E.
Yeah.
coli. The polar flagellum acts like a propeller, and it gives them this very directed, rapid movement.
And Jawas have that great description of it under dark field microscopy.
Shooting star motility, that's the one. Because they just zip across the field of view. It's actually quite beautiful, if you can momentarily forget that it causes devastating disease.
It makes sense for an aquatic organism. They need to be able to move around in their environment.
Absolutely. And speaking of their environment, we have to talk about salt. These organisms are halophilic.
Halophilic.
Salt-loving.
Right. Nearly every species in the Vibrio genus needs sodium chloride, NaCl, to grow. If you take Vibrio parahaemolyticus and try to plate it on a standard lab agar with no added salt, nothing will happen. It just won't grow.
But, and this is a huge but, there's one major exception. The king of the genus.
Vibrio cholerae. And this is a critical differentiator in Murray. Vibrio cholerae can grow on most media without extra salt. It likes salt, it grows better with it, but it doesn't strictly require it.
And that has massive epidemiological implications.
Huge. It's why V. parahaemolyticus is a coastal disease tied to saltwater. But cholera can contaminate a freshwater river or a well hundreds of miles inland and cause a massive outbreak. That's a great clinical clue. If the exposure is a freshwater lake, you should be thinking cholera or Aeromonas, not the others.
Exactly.
Now, even within Vibrio cholerae, it's not just one single bug. There's a whole naming system we have to get into. Murray says there are over 200 serogroups. That sounds like a memorization nightmare.
It does, but we can simplify it a lot. The serogroups are based on the O antigen. Remember, gram-negative bacteria have that lipopolysaccharide, LPS, in their outer membrane. The O antigen is the long sugar chain sticking off the end of it. It's like the team jersey the bacterium is wearing.
And out of 200 different jerseys.
Only 2 of them are associated with major epidemics, O1 and O139.
Okay, that's manageable. So if a lab report comes back with Vibrio cholerae O42,
that patient might have some mild diarrhea, maybe a wound infection if they were in the water, but they're not going to be the start of a pandemic. The ones that cause the massive, devastating outbreaks are almost exclusively O1 and O139. They have the genetic toolkit for it.
So O1 is the main villain. But Murray goes even deeper. He splits O1 into serotypes and biotypes. This feels like we're getting into the weeds, but I know this is prime exam material.
It is. Let's start with the serotypes. Within the O1 group, you have 3 main flavors: Inaba, Ogawa, and a third one called Hikojima.
Hikojima, that one always stands out.
It's pretty rare, and it's unstable. It's basically a transitional form where the bacterium expresses both the Inaba and Ogawa antigens at the same time. The bacteria can actually switch between these serotypes during an outbreak.
But does that matter for treatment?
Not at all.
Yeah.
Clinically, it makes no difference. You rehydrate them all the same way. The biotypes, though, classical versus L-Tor, that's a different story. That has real historical and epidemiological weight.
Okay, tell me about that.
The classical biotype is the one that caused the 6th pandemic. We're talking late 1800s, early 1900s. It was known for causing incredibly severe rapid disease. But it's mostly gone now.
It's been replaced.
It's been replaced by the L-Tor biotype. L-Tor is the cause of the 7th pandemic, the one we're technically still in, which began in 1961. L-Tor is interesting because it's a hardier bug. It survives much better in the environment.
And Jawitz gives us a few lab tricks to tell them apart.
Yep. If you're in a high-level lab, you can differentiate them. L-Tor produces a hemolysin, so it can lyse red blood cells. It's positive on the Voges Proskauer test. And the big one for exams, it's resistant to an antibiotic called polymyxin B.
So L-Tor is the tougher, more modern version.
And it has a really sinister advantage. It causes a much higher ratio of asymptomatic to symptomatic infections. So for every person who's desperately sick with L-Tor cholera, there might be 50 or 100 people walking around feeling fine, but shedding bacteria in their stool.
Which makes it almost impossible to control.
Exactly.
One last point on structure before we move on. The capsule.
Hmm.
Usually when we think of capsules in microbiology, we think of them as an invisibility cloak that lets bacteria get into the bloodstream.
That's a perfect analogy. And it holds true here. Vibrio vulnificus has a capsule, and so does the V. cholerae O139 strain. This acidic polysaccharide capsule protects them from phagocytosis. It stops our white blood cells from eating them.
And that's what allows them to cause systemic infections, like septicaemia.
Correct. But here's the twist. Vibrio cholerae 01, the classic pandemic strain, does not have a capsule.
Which tells you everything about its game plan.
Everything. V. cholerae 01 has no desire to be in your blood. It wants to stay right there in the lumen of your small intestine, stick to the wall, and become a toxin factory. It is a profoundly non-invasive pathogen.
That distinction, invasive versus non-invasive, is the perfect way to get into Section 2. Pathogenesis. Let's look inside this toxin machine.
This is one of the most elegant and terrifying stories in microbial genetics. Vibrio cholerae wasn't born a killer. It had to acquire its weapons. Murray describes this as a double-hit process.
Like a video game character finding power-ups.
Exactly like that. Power-up number 1 is a chunk of DNA called the Vibrio pathogenicity island, or VPI1. This island gives the bacterium the genes to build a special little appendage called the TCP.
The toxin co-regulated pilus. A pilus is just a little protein hair, right? So how is that a weapon?
Well, initially its main job is just adherence. It helps the bacterium stick to the gut wall so it doesn't get flushed away. But here's the crazy part. The TCP also happens to be the specific receptor for a virus.
The bacteriophage. The CTX phage.
Yep, a filamentous bacteriophage called CTX. So imagine this. The bacterium grows this little arm, the TCP, to hang on, but this virus comes along and sees that arm as the perfect docking station and doorway to infect the bacterium.
So the bacteria itself gets an infection.
It does. And the virus injects its DNA into the bacterial chromosome. And on that viral DNA are the genes CTXA and CTXB.
The blueprints for the cholera toxin.
The very blueprints. It's a beautiful, terrible example of coevolution. No pilus, no phage infection, no toxin genes, no toxin, no cholera.
That is just wild. The disease that's killed millions of people is basically the result of a bacterium catching a cold.
Pretty much.
Okay, so now the bacterium has the code and it's making the toxin.
Mm-hmm.
Let's break down how this toxin works. It's the classic AB toxin model.
Right. Think of it as a two-part weapon system. You have 5 B subunits that form a ring. B is for binding. This ring is perfectly shaped to lock onto a specific receptor on our intestinal cells called the GM1 ganglioside.
So the B subunits are the key, and the GM1 ganglioside is the lock.
Perfect analogy. Once the B subunits bind, they create a channel that allows the A subunit to enter the cell. A is for action. This is the part that actually does the damage.
And once that A subunit is inside, it goes straight for the cell's main control panel.
It does. It targets a regulatory protein called a G protein, specifically the stimulatory G protein, GS. Normally, GS acts like a light switch. It turns on, activates an enzyme called adenylate cyclase, and then quickly turns itself off. It keeps everything in balance.
But the toxin messes with that switch.
It breaks the switch. The subunit performs a chemical reaction called ADP ribosylation. It essentially welds a molecule onto the G's protein, which locks it permanently in the on-end position. It cannot turn off.
So adenylate cyclase just goes nuts.
It goes into overdrive. It starts churning out insane amounts of a signaling molecule called cyclic AMP or CAM-MP. And CAM-P is a second messenger. It basically starts screaming a set of instructions at the cell.
And in an intestinal cell, what are those instructions?
Disaster. The high levels of CAM-MP tell a specific channel, the CFTR channel, to start pumping chloride ions out of the cell and into the gut lumen.
So it's just dumping chloride.
Dumping chloride. And at the same time, it blocks other channels from absorbing sodium from the gut. So you have chloride rushing out and sodium being blocked from coming in. The gut becomes incredibly salty.
And then basic physics takes over. Osmosis.
Water follows salt. Because the ion concentration in the gut lumen is now so high, water from your cells and your bloodstream just pours across the membrane to try and dilute it.
The body is trying to establish equilibrium, but it's killing itself in the process.
That's exactly right. The result is this massive, unstoppable loss of fluid. We are talking up to a liter of fluid per hour in a severe case.
A full Nalgene bottle of water every hour.
Think about that. That's potentially 24 liters a day. That's completely incompatible with life unless you can replace it just as fast.
Which brings up a question. With this fire hose of fluid rushing out, why aren't the bacteria themselves just washed away?
That's where the TCP, the pilus, comes back into play. It's not just a receptor for the virus, it's also a super strong anchor. It allows the bacteria to clump together and adhere tightly to the mucosal surface. They just hold on and keep pumping out toxin.
But they don't dig in. They don't invade.
Nope. They stay on the surface. If you were to take a biopsy of the intestine during a cholera infection, the tissue itself would look surprisingly normal. There's no blood, no inflammation, no army of neutrophils like you'd see with Shigella.
Which explains the clinical picture we'll get to later. No fever, no blood in the stool.
Exactly. It's a toxin-mediated disease, not an invasive one.
Okay, let's switch gears to the other Vibrios. Section 3. Pathogenesis of V. parahaemolyticus and V. vulnificus. Let's start with the one you get from sushi, V. parahaemolyticus.
So parahaemolyticus also causes a watery diarrhea, but the mechanism is different. Its main weapon is a toxin called the thermostable direct hemolysin, or TDH.
Also known as the Kanagawa hemolysin.
Right, named after where it was discovered in Japan. And this leads to a very specific lab finding called the Kanagawa phenomenon. Pathogenic strains of V. parahaemolyticus, when you grow them on a special blood agar made with human blood, will show beta hemolysis.
Wait, it has to be human blood, not the usual sheep blood agar.
That's the key detail. Sheep red blood cells aren't susceptible. The toxin forms pores in human red blood cell membranes, causing them to lobines. In the gut, it doesn't cause CAMP to go up. It causes an increase in intracellular calcium.
Which also triggers chloride secretion.
Yes. Different pathway, same end result. Watery diarrhea, though usually not as severe as with cholera.
Okay, now for the really scary one. Vibrio vulnificus. You called it the aggressor.
And it absolutely is. Unlike cholerae, vulnificus wants to invade. We mentioned its polysaccharide capsule, which helps it evade the immune system. But the other critical factor, the one that really determines life or death, is iron.
Why iron? What's so special about iron?
All bacteria need iron to live, but our bodies are smart. We have a defense system called iron withholding. We keep almost all of our iron locked up tight inside proteins like transferrin, so bacteria can't get to it.
It's nutritional warfare. We try to starve them.
Exactly. But Vibrio vulnificus is a master scavenger of iron. Now, in a healthy person, there's very little free iron floating around for it to grab. But think about a patient with a condition like hemochromatosis.
An iron overload disorder.
Or more commonly, a patient with chronic liver disease, like cirrhosis from alcohol or hepatitis. A damaged liver can't regulate iron properly. The amount of free iron in their blood skyrockets.
So for the bacteria, the bloodstream suddenly becomes an all-you-can-eat iron buffet.
And that is precisely why these patients are at such extreme risk. A person with cirrhosis eats a raw oyster containing V. vulnificus. The bacteria get into their bloodstream, find this high iron paradise, and they multiply explosively.
And mortality rate for that primary septicaemia is over 50%, right?
Yes. It is a true medical emergency. Death can happen within a day or two of eating that oyster.
Which leads us perfectly into Section 4, epidemiology. Where do these things live? The simple answer is water, but it's more complex than that.
The ecology is everything. These are marine organisms. They live in coastal waters and estuaries, and they have a special relationship with zooplankton, particularly tiny crustaceans called copepods.
Little microscopic shrimp-like things.
Right. The Vibrio bacteria attach to the chitin in the copepod's shells. So when conditions are right, warm water, lots of nutrients, you get an algal bloom, which feeds the zooplankton. The zooplankton population explodes, and with them, the Vibrio population explodes.
So a cholera outbreak isn't just about sanitation. It starts with the marine ecosystem.
It does, but for humans to get sick, we have to ingest it. For V. cholerae, that's the classic fecal-oral route. You drink contaminated water or eat food prepared with it, but we have a powerful first line of defense, our stomach acid.
Acid barrier.
Vibrio cholerae is actually very sensitive to acid. The pH in a healthy stomach is low enough to kill most of them. So, to get sick, a healthy person needs to swallow a massive dose. Murray says over 100 million organisms.
That's a huge number.
It is. But what if you're taking a proton pump inhibitor, a PPI, for acid reflux? What if you have low stomach acid because of malnutrition?
Your shield is down.
Completely down. The infectious dose plummets by thousands of times. You might only need 10,000 organisms to get sick. So in an outbreak, people on antacids are at much higher risk.
That's a massive clinical pearl. Always ask about PPI use.
Absolutely. For the non-cholera vibrios, the vector is almost always seafood. Oysters are the classic culprit. They're filter feeders. They sit there all day filtering gallons and gallons of water, and they concentrate the bacteria in their tissues.
So an oyster basically becomes a little bag of concentrated Vibrio.
That's a great way to put it. And then we eat them raw, especially in the summer months when the water is warm and the bacterial counts are highest. It's a recipe for disaster.
Let's move on to what that disaster looks like. Section 5. Clinical diseases. You have the rice water versus the purple boule.
Let's start with cholera. Incubation period is usually 2 to 3 days. The onset is incredibly abrupt. There's no feeling sick for a day. You just suddenly start vomiting and having this massive, effortless, watery diarrhea.
Effortless is a truly terrifying word for diarrhea.
It describes it well. There's not a lot of cramping or straining at first.
Huh.
And the stool itself is characteristic. It's called rice water stool.
Why rice water?
Because it looks like the water you'd get after washing rice.
Yeah.
It's colorless, mostly odorless, and it's filled with these little flecks of mucus that look like grains of rice.
And at that point, it's not really stool anymore, is it?
No. After the initial bowel contents are cleared, the patient is just excreting what is essentially plasma ultrafiltrate. It's an isotonic fluid full of electrolytes.
And the systemic effects are all downstream from that fluid loss.
Dehydration causes sunken eyes, a loss of skin turgor. You get washerwoman's hands from the wrinkling. You develop a severe metabolic acidosis from losing bicarbonate and hypokalemia from losing potassium.
And the hypokalemia can cause cardiac arrhythmias.
Yes, and eventually you go into hypovolemic shock. Blood pressure plummets, the kidneys fail, and the patient dies. Untreated, the mortality rate is up to 70%.
But with treatment, it's less than 1%.
It's all about logistics. Can you get fluid into the patient faster than it's coming out? It's that simple and that difficult.
Now contrast that with V. parahaemolyticus.
You ate some questionable oysters. A day later, you get an explosive watery diarrhea. You have cramps, you feel nauseous. It feels awful, but it's almost always self-limiting. It resolves in about 3 days with just supportive care and hydration.
And then the horror show, V. vulnificus.
2 main presentations. The first is primary septicaemia. This is our patient with liver disease. They ate the raw oysters. They suddenly develop a high fever, chills, and then the skin lesions start.
Describe these lesions. This is a classic board question.
They're metastatic cutaneous lesions. The bacteria are seeding the skin from the inside, from the blood. It starts as a red, swollen patch, usually on the legs, and within hours it evolves into these massive, blood-filled blisters. Hemorrhagic bully.
And they turn black.
They do. They become necrotic. The tissue dies. It's a necrotizing vasculitis. It's absolutely devastating to see.
And the second presentation is a direct wound infection.
Right. And this can happen to anyone, even a healthy person. You're walking on the beach and cut your foot on a shell, or you're handling seafood and cut your hand. The bacteria get inoculated directly into the wound. You get swelling, redness, and then that same rapid progression to bully and tissue necrosis.
Okay, section 6, laboratory diagnosis. How do we confirm which one it is?
Well, first, you have to be careful with the sample. Vibrios are delicate. They can die if the stool sample dries out or gets too acidic. Carrie-Blair is the recommended transport medium.
When it gets to the lab, the key is the special agar, TCBS.
TCBS, thiosulfate citrate bile salts sucrose agar. It's a mouthful, but each part is important. The bile salts and high pH inhibit most other gut bacteria, but the key ingredient for differentiation is the sucrose.
This is the traffic light system.
Exactly. The agar has a pH indicator in it. If the bacteria can ferment sucrose, it produces acid, which turns the colonies yellow.
And Vibrio cholerae.
Ferments sucrose. So V. cholerae gives you bright yellow colonies on TCBS.
And the others?
V. parahaemolyticus and V. vulnificus do not ferment sucrose, so they don't produce acid. The colonies stay green, which is the original color of the agar.
Simple.
Yeah.
Yellow for cholera, green for the bad cousins.
That's the mnemonic. And one other trick. If you think there are very few organisms in the stool, you can use an enrichment broth called alkaline peptone broth. It has a high pH, around 8.6, which Vibrio loves, but most other bacteria hate. It gives the Vibrio a head start.
All right, section 7, treatment. We've said it again and again, but let's hammer it home. The number one priority is
FLUID. Rehydration is everything. And we have to talk about one of the greatest medical inventions of the 20th century, oral rehydration salts, or ORS.
It seems so incredibly simple. Just a packet of salt, sugar, and electrolytes you mix in water.
Simple, but physiologically brilliant. Remember how the cholera toxin blocked the normal sodium absorption channels?
Right.
Well, it turns out it doesn't block a different channel, the sodium glucose cotransporter, or SGLT1. This transporter pulls one molecule of glucose and one molecule of sodium into the cell together.
It's like the toxin blocked the front door, but ORS uses the side door.
That's a perfect way to describe it. By giving glucose with the salt, you bypass the toxin's blockade and trick the gut into absorbing sodium again. Water follows the sodium, and you rehydrate the patient using their own gut. It's incredible.
And for patients in shock who need IV fluids, we use Ringer's lactate. Why not just normal saline?
Because the patient is profoundly acidotic from losing all that bicarbonate in their stool. Ringer's lactite contains lactite, which the liver metabolizes into bicarbonate, helping to correct the acidosis. Normal saline would actually make the acidosis worse.
What about antibiotics?
For cholera, they're secondary. A single dose of doxycycline or azithromycin will decrease the total volume of diarrhea and shorten how long the patient sheds the bacteria. It helps with public health and control, but the fluids are what save the patient's life.
But for Vibrio vulnificus, that's a different story.
Oh, completely different. For vulnificus, immediate and aggressive antibiotics are life-saving. We use a combination, typically a tetracycline like doxycycline, plus a third-generation cephalosporin like ceftriaxone, and often emergent surgery. You have to debride all that dead necrotic tissue. Sometimes amputation is the only way to stop the infection from spreading.
Okay, before our quiz, let's quickly cover the imposter. Aeromonas, Section 8.
Right, Aeromonas. This is the freshwater cousin. It's also an oxidase-positive, gram-negative rod, so it can look like Vibrio, but its habitat is rivers, lakes, even contaminated tap water.
And a key difference is its antibiotic resistance pattern.
Yes. Aeromonas is often resistant to penicillins and many cephalosporins. You usually have to use something like a fluoroquinolone.
And for exams, there are 3 classic clinical associations.
Number 1 is just a standard diarrheal disease, especially in kids or people who drink untreated water. Number 2 is wound infections from freshwater exposure, a fishhook injury, a cut while water skiing.
And number 3 is my favorite weird microbiology fact. Medicinal leeches.
The leeches, yes. So medicinal leeches are still used in reconstructive surgery to help with venous congestion and skin flaps, but the leeches have a symbiotic bacteria in their gut that helps them digest blood.
And that bacteria is Aeromonas.
It is Aeromonas varroni. So the leech bites the patient and it can regurgitate some of its gut contents into the wound. If a patient who's had leech therapy develops a wound infection, it's Aeromonas until proven otherwise.
That is just fantastic. Okay, we have navigated the murky waters. Now it is time for our rapid recall to see what's stuck.
All right. I'll pose the question. You at home, pause, think about the mechanism, and then we'll break it down.
Question 1. The cholera toxin works by permanently activating a G protein, leading to the massive overproduction of which specific intracellular signaling molecule? The answer is CAMP, or cyclic adenosine monophosphate.
Right. It's the constant activation of adenylate cyclase that leads to the huge buildup of CAMP. That's what drives the chloride secretion and the catastrophic water loss.
Question 2. You're looking at a TCBS agar plate from a stool sample. You see large, bright yellow colonies. Which organism is this most likely to be, and what biochemical reaction is causing that color change? That would be Vibrio cholerae. And the colonies are yellow because it ferments sucrose.
Exactly. Sucrose fermentation produces acid, which drops the pH, and the pH indicator in the agar turns yellow. V. parahaemolyticus would be green because it doesn't ferment sucrose.
Question 3. A 62-year-old man with a known history of alcoholic cirrhosis comes to the ER with fever, shock, and rapidly developing large, blood-filled blisters on his legs. He mentions eating raw oysters the day before. What is the most likely pathogen? That has to be Vibrio vulnificus.
Without a doubt. The combination of liver disease, raw oyster consumption, and those classic hemorrhagic bullae is the textbook presentation for Vibrio vulnificus sepsis.
Question 4. A gram-negative comma-shaped rod is isolated from a patient with watery diarrhea. It is oxidase positive and grows in saltwater environments. A key structural feature is that it does not possess a polysaccharide capsule. Which specific organism are we talking about?
Yeah.
This has to be Vibrio cholerae 01.
That's the key. It fits the Vibrio profile, but the lack of a capsule is what separates the 01 pandemic strain from V. vulnificus and V. cholerae 0139, both of which are encapsulated and more likely to be invasive.
Last one. Question 5. A patient develops a serious wound infection after a traumatic injury in a freshwater lake. The cultured organism is an oxidase-positive rod that is resistant to ampicillin. What is the most likely cause?
Aeromonas.
Perfect. The freshwater exposure points away from the marine Vibrios. The oxidase-positive result separates it from enteric bacteria, and the resistance profile is classic for Aeromonas.
Excellent. If any of those were tricky, that's your cue to rewind and listen to that section again.
And just one final thought to leave you with. We mentioned it briefly, but let's call it the climate connection.
Right. Murray specifically calls out that Vibrio parahaemolyticus outbreak in Alaska back in 2004.
Think about that for a second. Alaska. Vibrio was always considered a disease of warm, tropical waters, but cruise ship passengers got sick from eating locally harvested Alaskan oysters because the water temperature in Prince William Sound had crept up just enough, above 15 degrees Celsius, to allow the bacteria to bloom.
So it's not a tropical disease anymore. Its range is expanding. As the oceans warm, that Vibrio belt is moving north and south from the equator. We're going to start seeing these infections in places we never would have expected them before.
It's a stark reminder that microbiology and clinical medicine don't exist in a bubble. They're completely intertwined with the environment and the health of our planet.
Exactly.
On that rather sobering note, stay safe, make sure you cook your shellfish, and always respect the power of electrolytes.
And keep studying.
We'll see you in the next deep dive.